Okay, well, thank you for this invitation to come here today and speak about the work that we've been doing. So I'm a toxicologist, but I'm also a molecular cytogeneticist, and I was involved in some of the CNV discovery projects that earlier in my career. And so the question that we're asking in my lab and that we're interested in is looking at the role of chemical exposure in generating CNVs. So I'm going to talk today about how we're going about doing that, and also talk about how we're using our model, the zebrafish pictured here, to go about answering this question. So we've had a wonderful introduction already for this session, so I'm not going to have to spend a lot of time on my intro slides, but I'll just spend a few seconds on each of them, kind of highlighting maybe some of the points that may not have been covered. So we are talking about CNVs as just one of the types of genetic variation that we know that we have within the human genome, and it was only recently that we've had this discovery, and since that time we've had a number of other additional studies that have been conducted now to refine the CNV maps and try and understand the implications of these CNVs within the genome. And Jim gave a wonderful overview of some of the diseases that CNVs are associated with, and especially looking at the studies where we're looking at these spontaneous CNVs and the different disease associations that have been associated with those spontaneous CNVs, including autism, schizophrenia, and some other Alzheimer's disease and other diseases that he talked about earlier today. So beyond humans, there's been a number of other studies that have been conducted in other vertebrate models looking at CNVs and doing CNV maps in those models. And for the most part, every model that has been looked at to date, they've been able to find CNVs within those genomes. There have been a number of models that have been investigated, and I've just included a few here, such as the primate studies, cattle, pigs, dogs, rodents, and the model that we use, the zebrafish. So as I mentioned, the question that we're going about addressing in our lab is, does chemical exposure induce CNVs? And I just wanted to give a little information of why I think this is important, and starting off with some about what we know about in the CNV world right now. So we know that CNVs play a role in complex diseases, and we've talked about that throughout the session today. But what we don't know is what environmental stressors are involved in generating some of these spontaneous CNVs. We also know that a lot of these complex diseases are influenced not only by genetic factors, but also by environmental factors. But what we don't know is what exactly these environmental factors are. And there's a lot of ongoing studies that are trying to link a lot of the different environmental stressors and with these different complex diseases and understand the influences of these environmental exposures. So from the environmental stressors, since I'm a toxicologist, our interest is then looking at environmental chemical exposure and putting these two together and figuring out how an environmental chemical exposure and their role in potentially generating CNVs. And the overarching reason we're doing this is trying to add, I see really putting this copy number variation as really just another piece of the puzzle when we're trying to understand all of the different factors that are contributing to these complex diseases. And so hopefully by understanding and answering this question, looking at how environmental chemical exposure generates spontaneous CNVs, we can also add more of our information into understanding the mechanisms that are influencing these disease onset. So why has this not been investigated before? And I think Jim actually led me into this, answering this question for him, because he was talking about the Ames assay and whether or not the Ames assay would be able to detect CNVs. And my point of view is that we really, so far in a lot of our studies where we're looking at environmental chemical exposures, we've been on two different poles. We start up here, we're looking at a lot of the mutations, the single nucleotide changes, where we have a lot of good genotoxicity assays that are developed and we've been able to use throughout these studies. But then we also have on the other end of the spectrum the cytogenetic assays, where we're looking at a lot of these larger microscopic alterations, where we can do a lot of the chromosomal assays looking at breaks and things under the microscope and being able to detect them. But when we're talking about the copy number alterations or the copy number variants, what we have here is we're looking in the middle. And it really hasn't been until recently that we've had these technologies and assays that are out there where we can efficiently look at the whole genome and really start to ask this question of whether or not chemical exposure might be will be causing CNVs. And it's only been since, as I mentioned, 2004, where we've known we've had this widespread copy number variation in phenotypic normal individuals, and they really beginning to understand the influence of the spontaneous CNVs on the complex diseases. So a couple of the assays that we have available to us in technologies, there's been touched on already before a little bit about array comparative genomic hybridization, and then also whole genome sequencing. So we're going about answering this question using a combination of these assays in our lab, using array CGH and also whole genome sequencing to ask this question and look at whether or not chemical exposure induces spontaneous CNVs. Just a quick overview of what these assays and how we're going about using these assays to detect these changes. Array CGH, in case anyone is not familiar with what array CGH is, it's very similar to the gene expression microarrays, except for looking at the gene regions. We're interested in looking at the genome-wide view. It's a comparative analysis where we have our reference sample labeled in one color and we compare it to our test sample. We co-hybridize those on a microarray, and then we look for changes in fluorescence intensity. And so if we have this equal number of copies within the two genomes, we expect to see this equal 1-to-1 ratio, and we would plot it right here along the line. But if we have a loss in the event, we get a negative deviation from that ratio, or a gain event occurring, we get a positive deviation from that ratio. And so we can go through the genome and mine the genome and look for those copy number events that are occurring. And using the array CGH method, our resolution is ultimately dependent on the size and the density of our genomic elements that we have spotted on our arrays. There's a lot of different commercial array platforms that are now available, and also commercial software that is out there. So we can do these assays quite efficiently in the laboratory. Another genome-wide assay that we can use is whole genome sequencing. Now that we're available to do these high throughput sequencing approaches, we can look at the whole genome. And these specific methods are based upon the specific sequencing approach that you're going to take, but it involves isolating your genomic DNA, preparing your template, doing the sequencing, and then doing your complex genome alignment and your comparative genome analysis to get your answers. So as I mentioned in the beginning, we are going about asking this question using the zebrafish vertebrate model system in our laboratory. We're coupling both an in vitro and an in vivo approach, similar to how Tom's doing with his experiments with his cell culture and the mice. And what we're really looking at is trying to identify these CNVs and then really looking at the functional impact and using the in vivo exposure to really try and understand more of this functional impacts if we're getting these spontaneous CNVs that are occurring following the chemical exposure. So in case you're not familiar with the zebrafish model system, I want to spend a little bit of time just talking about some of the strengths and a little bit about the model system itself. The zebrafish is a small tropical fish that's native to eastern India. It's been used in biological research, developmental biology research, for quite a few years, since the 1930s. The 1970s, the model began to gain popularity when they were looking for an inexpensive vertebrate model system for forward genetics. And if you go and you look through the literature today, from about the 1990s to today, we see a wide expansion of the use of this model in almost every area of biology. And currently, probably if there's a certain disease, there's probably a zebrafish model right now out there to begin to interrogate and understand those disease pathways. So some of the strengths that we're taking advantage of for our question that we're asking is one is the hundreds of eggs that the zebrafish are able to produce weekly. So we can put together an adult male and adult female fish, breed them together, and we can get hundreds of eggs from just that single mating pair. And so we have plenty of organisms that we're able to use and that are available to us in the laboratory. So you can imagine if we have multiple breeding pair sets, how many actually individuals that we can start to work with for our assays. We also take advantage of their ex utero embryonic development. So the females will lay their eggs, the males will fertilize that. It's all done externally. So we're able to go in and collect these fertilized eggs and put them in petri dishes to do our chemical exposures. We don't have to worry about dosing, the parents at all for these chemical exposures to be able to do these exposures during this embryonic development. The embryos are singular and they're transparent, so we can go and look under a light microscope and actually look at the developing embryos under the light microscope and look for any type of phenotypic changes or morphologic changes that are occurring within the fish. They have short developmental periods, so we can look at the whole developmental period within about 3 to 4 days if we're interested in doing an exposure during the developmental embryonic period. They're sexually matured about 3 to 4 months, so if we're interested in doing multi-generation studies, we can get those multiple generations in just a few months. Their lifespan is a few years, so it's a fairly quick lifespan. They're a small-sized adult organism, so we can house a lot of individuals in just a small amount of space. They're about 1 inch to 1.5 inches total in size, and they are amenable to genetic manipulations. The zebrafish does have a finished genome sequence, so this is good for us when we're doing the CNV studies. They have 25 pairs of chromosomes. You notice from the ideogram here The chromosomes are actually not labeled similar to what you would see in most mammalian karyotypes, and that the largest chromosome is not 1 and the smallest one with a higher number. Instead, right now for the zebrafish chromosomes, they're just transferred from the linkage group number that they were originally assigned when we're talking about the specific numbers for the zebrafish chromosomes. The genome size for the zebrafish is about half the size of the human genome, and I've been involved in the studies that we were doing, the cytogenetic mapping and characterization of the genome. So one key element that we want to remember is that gene function is conserved among vertebrates. Zebrafish are a vertebrate, and so that our hope is that the findings that we see, that what we find in the zebrafish will be able to translate, hopefully, over into humans. It's estimated about 80% of the zebrafish genes are homologous to human genes, and there's been a number of studies doing mutations in different zebrafish genes, and these mutation events display similar phenotypes that they would see in human diseases. So further confirming that these genes are function is similar down into the zebrafish as they are for humans. So before we could embark on asking our question and looking at whether or not chemical exposure would induce CNVs using the zebrafish, we recently completed a CNV map for the zebrafish. This was a study that is a collaborative study with the laboratory of Dr. Charles Lee at Brigham and Women's in Harvard Medical School. And for this study, what we did is we took 3 different wild-type zebrafish strains that are used in laboratory studies, the A, B, WIC, and 2 strains. We took 10 individuals, and these 10 individuals were collected, 10 from each of 2 different labs so that we wouldn't have a lab bias that was going on. And we looked at their genomes and created CNV profiles using the array-CGH technology. We also collected 10 individuals from 2 different sites in India to get a CNV profile for the native fish too, and compare those to the laboratory strains. So from this analysis, we identified over 31,000 different CNVs, but we went in and refined our number of CNVs into what we called CNV elements, where we went through and we looked for regions that we had overlapping CNVs, such as shown here, and we classified that into a single CNVE. And we had over 6,000 CNVEs with a mean size of 41 kb and a median size of 23 kb. So this image here is just showing you all of the different CNVEs that we detected within the zebrafish genome. I'm not going to spend a lot of time going through and all the detailed analysis of this about CNVs within the zebrafish genome, but really what I just wanted to show you is that the zebrafish genome does have CNVs, and so we find that it would be a suitable model for us moving forward with asking our question. Yes?
Sorry, doesn't the zebrafish genome have a lot of genome duplication? Can you distinguish your
Right. Right, that is true. It is duplicated. The zebrafish has to have a duplicated genome, and we can go through and separate that out.
Okay.
Yes.
There's enough information.
Yes.
Thank you very much.
So I want to spend the rest of my time talking about just our proof of principle experiment that we've been doing with a known genotoxic chemical to answer and begin to answer this question before we've been able to move into different environmental chemicals. We've been using a known genotoxic chemical, ethyl methane sulfonate, or EMS, which is known to induce mutations to begin to get at our question of whether or not chemical exposure will induce the spontaneous CNVs. For this initial study, we used our cell line that we had available in the laboratory, and we wanted to start in the cell line and then move into in vivo just because of the ease of really starting in the cell line versus moving into, before we move into the in vivo model, to first get a screen and an understanding of whether or not we would see CNVs from these specific chemical exposures. We included multiple doses in this experiment. We included one where we had no cytotoxicity, and then we also included doses where we had some cell death occurring, just to get some comparison of what type of CNVs, or if we would see CNVs at all those different doses when these different events were occurring. So for this initial analysis, we used the array-CGH technology. We used our control, our no chemical treatment, as our reference sample, and then we co-hybridized that with each of our different chemical treatments, and then analyzed that with the array-CGH so we could detect and get our CNV profiles following the chemical exposure at each of these different treatments. So what we saw is that we were seeing spontaneous CNVs that were being generated following this chemical exposure. We were seeing CNVs at all of the different chemical concentrations that we used. And this figure here is showing the location. We are seeing them on multiple different chromosomes. We are seeing both gain and loss events that were occurring. And one of the interesting things to me is that when we looked at the multiple concentrations, we did see similar genomic regions that had harboring these spontaneous CNVs at these multiple exposure doses. And I just included one here where we saw a gain event occurring on chromosome 14. It ranged a little bit in different sizes from 126 to 173 kb in size among the different concentrations. But we were seeing a similar region in all of our different concentrations that we were seeing these spontaneous events occur. We really weren't seeing a dose response event at all either in our studies when we were doing these different chemical exposures, so I just wanted to point that out. The next question that we wanted to get at, that I wanted to switch to, is whether or not, one of the questions we get, okay, what is the biological significance of seeing these spontaneous CNVs? Is there any biological function or biological alterations that you might be seeing? So the next step that we are taking is looking at whether or not we're seeing changes in gene expression. And so what we are starting to do to get at this question is that we had our cells that we treated either with our control treatment or our different chemical treatments. And remember, we took we isolated DNA from those samples to be able to do our race-CGH analysis. But we also isolated total RNA from these samples too. And we conducted global gene expression using a zebrafish microarray platform that contained all the different zebrafish genomes to begin to see whether or not we would see gene expression changes and what type of gene expression changes we would see, and begin to correlate the gene expression changes with the areas where we were seeing our spontaneous CNVs. And overall, what we're seeing is that we do see that the spontaneous CNVs correlate to altered gene expression, and that just doing the basic analysis of going through and looking at where these altered genes are located within the genome and whether or not they're located in these regions where we're seeing these spontaneous CNV events occurring. And so I included one example here in my presentation today. So we saw a 345 kb loss on chromosome 21 was one of the events that we were seeing. Following the global gene expression analysis, what we found is one of the genes that we saw downregulated was the CCR4 NOT transcription complex subunit 6, or CNOT6. And this gene is located within this region where we're seeing this loss. And so we're seeing a downregulation of a gene where we were seeing a loss, and we could correlate those two regions. If you go through and do gene ontology analysis and find out the function of this gene, you'll find out that CNOT6 actually targets DNA damage proteins and is required for cell cycle progression and prevent cell death. So it kind of makes sense of why you might see it from why you might see this alteration from exposure to a genotoxic chemical. And so what this is showing is just giving an example of how we can start to go in and look at whether or not these spontaneous CNVs and do they alter gene expression and what their biological significance might be. So we've started doing a lot of the in vitro work, and now we're moving into starting our in vivo analysis. We're really at the early stages of this analysis, but I want to spend just to give a little bit of time about what we're doing and how we're going about this, and what strengths we're taking from the zebrafish model to go about answering this question. So I mentioned we can mate a single male and female pair, and we can get hundreds of fertilized embryos from this pair to be in. So what we're doing is we're taking those embryos and we're dividing them up from that single pair among our control or our different chemical treatments. We're exposing those embryos through developmental periods, for the first 72 hours of their life, and then we're doing their genomic DNA isolation, and then getting the CNV profiles from those individuals, so that we can compare the CNV profiles between the exposed and compared to the unexposed individuals. We're also taking it one step further and getting CNV profiles from the parents. We can get a tail fin clip from the parents and create their CNV profiles. The zebrafish have a regenerative caudal fin, and so their fin will regenerate. And so we're able to take clips of the fins and not harm the fish, but be able to get these CNV profiles and do this comparative analysis between that exposed population and also looking at the profiles from their parents. And so we're doing this to be able to look at one of the most sensitive periods, this developmental period, and seeing whether or not these chemical exposures will generate these spontaneous CNVs during this developmental period, which would be somewhat similar to the in utero fetal developmental period in humans. And we're working to identify the spontaneous CNVs that we get from these, and we're also want to move forward with looking at the biological and functional significance from this. One of our plans, what we want to do is also take from the individuals from these treatments. We will take some of them from the treatments and also grow them up throughout their lifespan to look and see whether or not what their influences are, and begin to sample them throughout the lifespan. And we're doing a similar approach, really from this kind of developmental origin of adult disease, which we talked a little bit about earlier today, looking for how these developmental exposures can influence or alter our potential influence disease onset later in life. So just in summary from what I've covered today, we've been able to complete a proof of principle experiment confirming that chemical exposure with that known genotoxic chemical will create spontaneous CNVs, and it also results in altering gene expression. But we're really just getting started in a lot of these studies. What we need to do next is really get into really being able to assess a lot of the environmental chemical contaminants and to see whether or not exposure to these chemical contaminants might also create spontaneous CNVs. And we need to get in and really do additional characterization of all these genomic element regions where we're seeing these spontaneous CNVs, so we can get into more of the mechanisms in which they're being created, and also working forward to looking at the functional significance of the alterations that we're seeing. So why is this important? As I mentioned earlier in the talk, we know that complex diseases are associated with spontaneous CNVs, and we also know that complex diseases are influenced by different environmental factors, but we haven't been able to combine the two, and we're really trying to understand the mechanisms that are influencing those diseases, and so we're looking at the spontaneous CNVs as just one of the pieces of that puzzle of how disease onset is influenced by chemical exposure. I just want to wrap up with acknowledging individuals in my lab that have contributed to this work, especially Dr. Sam Peterson, who is involved in a lot of this work, and acknowledge the laboratory of Dr. Charles Lee, who we are collaborating with the CNV map studies for the zebrafish, and two of the postdocs working in his lab, Dr. Kim Brown and Dr. Kim Dobrinsky, and also all the co-authors on our CNV zebrafish paper. And also want to acknowledge Dr. Len Zahn and Dr. Yi Zhao, who were responsible for creating and working with designing the gene expression microarray that we've been using in our studies. Thank you for your time, and take any questions.
So that was wonderful. I think this is a great model system. A few questions.
Okay.
Detailed. I think it is.
Okay.
Yeah.
Just not
So 2 questions about details of the experiments. Have you looked at breakpoint junctions, and where are you seeing? Are these both the normal and those that you induce? Are they being driven by homologous repair mechanisms or other?
So we're doing that right now. These are, we haven't done a lot of the detailed characterization yet of the genomic regions, and that's what we're ongoing right now. As I said, a lot of this stuff is work that's in process, and so we just don't have an answer yet.
I was going to bring it up in the discussion period, but I'll say now, I mean, clearly one of the challenges in choosing model organism systems in this is that what you see is dependent on genomic architecture and the repair profile of the cell, the repair capacities. If you look for CNVs in yeast following replication stress, you're going to see TY recombination events, mainly deletions, which is completely different than what we're seeing in mammalian cells. So I think that's an important thing, and that what's driving the kind of mutation may be the same up front, but what you see at the end may be different depending on the model system. So I think that's important.
So I just want to make a quick comment. For the CNV mapping studies that we did when we were looking, we did do breakpoint sequencing and characterization for those, and those were following similar mechanisms to what we're seeing in mammalian cells. We haven't done that detailed characterization yet for the ones that were generated by the chemical exposure.
And just a second real quick. So the in utero studies are going to give you mosaicism for CNVs in the offspring, right? Yeah. And you're starting out with a already duplicated genome. So the question is, you already have a duplication, you have 4 copies of chromosomes, and you have 1 CNV event, which is bringing it down to 25% of the copies of DNA. And then you're looking for mosaicism on top of that. So, you know, That's challenging. I mean, what sort of mosaicism do you think you'll be able to detect? What level?
It is challenging, and I've been listening today talking about all the single cell analysis that's been going on, and
what do you look
from
at?
the principal experiments, I mean, that's one of the questions I get quite often. We are detecting, but I mean, when we're looking at the results, we are detecting these spontaneous CNVs, and so I guess that's my counter argument. It is going to be complex. We will probably miss the lower penetrant one, definitely, but moving forward, I think we're going to be able to detect those ones that we're seeing at a higher rate and be able to detect those and, I guess, get a starting place for answering these questions.
So do you think you're preferentially detecting the ones that are occurring at the 1-cell or 2-cell stage?
Probably yes.
Very nice, Jennifer. Tom stole most of my questions, but one was, you said you did the CNV map and you characterized the breakpoints there, and it looked the same. How about some numbers? What was the frequency of microhomology at breakpoints? What was the frequency of finding pseudogene combinations or NEHR type mechanisms? Can you give us some insight?
If you want some exact numbers, we can talk about those later. Maybe that's.